It's just that the time has come because of these massive advances in biologic science and the ability to handle and work with these enormous data sets due to advancements in computational ability and advances in data science and machine learning and artificial intelligence, combined with having sequenced the human genome. We're at an incredible inflection point where the way healthcare will be delivered in the future.
Welcome to the Precision Medicine Made Simple podcast, where we simplify the complex world of precision medicine. We're here to explore how personalized healthcare can empower you to understand and improve your health to live a longer and healthier life. Join us as we break down the latest in medical science into actionable insights. Let's make precision medicine accessible and actionable for everyone. Enjoy the show. Today's episode is sponsored by Biography Health, where healthcare meets personalization. With their DNA-enabled blueprint, Biography Health empowers you with a health plan based on your unique DNA, aiming to prevent disease and extend your life. Take control of your health journey with a plan as unique as you. Visit biographyhealth.com and start your personalized healthcare journey today. As a reminder, this episode is for educational purposes only. This episode is in no way a replacement for professional health advice. Please consult with your professional primary care provider for medical advice. If at any point you feel that a topic discussed on our podcast was misrepresented, or if you have additional insights that could enhance our discussions, we wholeheartedly encourage you to reach out to us. Your feedback is invaluable in helping us improve and in ensuring our content remains both informative and reflective of the latest developments in precision medicine. These next two episodes are a two-part series to Dr. Stanford's presentation called Optimizing Healthspan and Longevity by Implementing Personalized Precision Medicine. Think of this episode like Precision Medicine 101. Dr. Stanford dives into a number of different topics, including the evolution of personalized precision medicine and how it's changing healthcare, the significance of knowing your risk by looking at genetics, family history, monogenic risk, and polygenic risk scores, and pharmacogenomics, which is tailoring drug treatments based on an individual's genetic variations. While this episode is available on audio platforms, we highly recommend watching this episode on YouTube, as Dr. Stanford references numerous visuals and graphs.
I'm excited today to share a talk with the listeners that is more of an educational, just like the entire podcast that we've been doing, intended to be educational. This is meant to take a little deeper dive into the science behind some of these things that we do when I talk about personalized precision medicine.
Awesome. Yeah, I'm excited to dive in, and I can imagine it gives more of this very general, but then also detailed, holistic view of what you've been doing for some time now.
Yeah, I'll crank in some case examples and stuff as we go.
Nice.
Today we're going to talk about personalized precision medicine. What is it? Why now? And how can you use it to optimize your health span and longevity? Oh, there's my disclosures. Major disclosure that I'm founder and chief medical officer of the Beverly Hills Institute for Precision Medicine, which is a practice that is involved in implementing personalized precision medicine. And I'm co-founder and chief medical officer for Biography Health, which is a tech platform. And our podcast is sponsored by Biography Health, a tech platform which is AI ready, that aggregates and analyzes precision medicine and other internal medicine type information into a way that provides interesting dashboards for patients and interesting dashboards for physicians to accomplish some clinical decision support. I'm on the speakers bureau for the Grail company, which makes the Gallery test, the multi-cancer early detection blood test, so-called liquid biopsy. And I'm also a speaker for GeneType, which is a company that takes genetic information and makes integrated risk scores. So this slide is a problem statement. Right now, on the left side of the screen, we're talking about the healthcare system as it exists now. It's reactive. It focuses on treating disease after it's already occurred. It is at population scale, one size fits all, guideline driven, doesn't account, and it doesn't really make an attempt to individualize the patient and understand them so you can get at the underlying causes, which reflects back on this focus on treating disease after it already occurs. The current healthcare paradigm focuses on symptom relief or getting at short-term fixes and improvements. So say, for example, with diabetes, if your hemoglobin A1C is 5.8, it's, oh my gosh, you're pre-diabetic. If you get it down to 5.7, which is virtually the same number, no one cares anymore. So it's very algorithmic like that, and again, guideline driven. Most of the people that encounter the healthcare system in the US as it sits today are not impressed. It's a industrial factory assembly line type model. We're also fairly hampered in our current healthcare system by the regulatory bodies like the FDA, CDC, CMS, which those organizations are based on protocols that were developed many years ago. They're fairly slow to adopt new technologies and medical advances. And when I say fairly slow, I mean the number you most often hear quoted is that from the time that something is found in the laboratory or in a study until it's been studied thoroughly enough to determine its safety and efficacy and make its way into general practice and guidelines and insurance reimbursement. That number, take a guess in your head, you'll be surprised, 17 years is what that takes. And by the way, Taylor, if at any point you have a question or you want some point of clarification, feel free to jump in because this is meant to be interactive. So on the right, what personalized precision medicine does and what would work better, what people really want, is a focus on prevention of disease, or at least delay the onset of chronic diseases. And if a chronic disease begins to occur during that transition period between health and disease, we want to detect and diagnose that disease at the earliest possible moment when it's easiest to treat. So for example, with diabetes, if you can catch someone in the insulin resistant stage, even before they're pre-diabetic, you can guide them such that they'll never become pre-diabetic or diabetic. And we know how to do that. And with cancer is most easily treated in its early stages at stage 1 or 2. Most of us intuitively know this, but there's science behind it. There's statistics behind it also. And it's very difficult to treat once the cat is out of the bag, so to speak, and it's metastasized to different parts of the body at this stage 4. What personalized medicine seeks to do is to treat each patient uniquely. We look at each person's points of genetic variation, their life history, exposures, their past medical history, their extended family history. All these together allow for the patient to be what we term personalized or individualized. What we're seeking to do is extend Healthspan. That is the period of your life where you have high functional capacity right up to the end of your life. The way things have gone over as long as any of us can remember is that human beings tend to reach their peak and around 40 years old or so in terms of their functional capacity, their cognitive ability, their athletic ability is probably more like around 25 years old. And then it's a slow, steady decline, picking up speed after about age 60 or 65. So not infrequently, the last decade or two of people's lives there is filled with infirmity and decreased functional capacity. In the current paradigm, patients are not really able to participate in their own care the way they would like to. And personalized precision medicine and the new healthcare paradigm that we're working toward would be a patient-centered experience where they're quite involved in their care. They're using wearable technology, they're self-quantifying, they're taking responsibility for their diet, exercise, etc. And they're quite involved in the process. That would be a much more effective way to go about things. And then in terms of the regulatory bodies, we want, and I've talked to people at the FDA and CMS, and they're aware that they need to completely change the way that they evaluate and we achieve adoption of new advances. So a good example is this. I mentioned briefly the Grail and Gallery test. They're talking about having a 3-year study to determine what is going to be included in their 7-year study to decide if that technology should be wheeled out broadly across the population. That in 10 years, the current technology along those lines of liquid biopsy will have completely advanced into other realms. So we're going to need to really just fundamentally change the way that we go about evaluating things in this time of just rapidly accelerating, not just rapidly advancing, in terms of the velocity of change, but the acceleration of change is just crazy. And of course, a lot of that driven by AI. But personalized precision medicine is not just a tweak on what we're doing. It's a completely different healthcare strategy. And this why now? And the why now is sequencing of the human genome, which completed in 2003, took 13 years at a cost of $3 billion. And even a few years ago, I say a few years, maybe 10 years ago, it probably still cost $100,000 and took months to get sequenced. Now it can be done in a day or two at a cost of certainly under $1,000 and sometimes even cheaper than that. In addition to that advance, there's also epigenetic sequencing that can be done. And we're going to talk about epigenetics quite a bit later on, about exactly what it is and how that can be leveraged in this personalized precision medicine. But suffice to say that genetics get at what your unique genetic variation is, and epigenetics gets at what is unique about you in terms of which genes are turned on or turned off at any particular moment. So you can do this genetic and epigenetic sequencing really quickly, low cost, and it's a fairly simple, highly repeated, accurate process. And there's a company out there called Oxford Nanopore, which has flow cell technology. It's not a commercial for them, but just to mention that they have a technique where you can do the genetic sequencing and epigenetic sequencing simultaneously. So it's very exciting with a device that will likely find its way into doctors' offices. So the lab to do that, I think in the future you won't even be mailing off the specimens to distant labs. You'll be able to do it right in the doctor's office. Starting about 50 years ago, most of you are not going to be old enough to remember this, but a former President Nixon had declared war on cancer. It was back in about 1972 or 73. And this spurred a lot of investment by the National Institute of Health and other scientific bodies in terms of really trying to understand from a basic science process what were the normal intracellular mechanisms and pathways, and just really trying to get at the underpinnings of what's going wrong at the cellular and molecular level to understand how diseases were occurring. This has slowly, over time, paid big dividends. I think back with the war on cancer, their goal was to cure cancer in like 5 years. Whoops, that didn't happen. But what they did learn is how complex of a disease things like cancer is, and how many different pathways are involved. Hundreds or thousands of different pathways that can go awry to lead to malignancy. And really understanding, and similar with dementia, these very complex things. It's taking decades and decades to start to untangle the spaghetti of what is normal physiology and what's going wrong to cause disease. Big boost in the last 5 to 10 years with the emergence of machine learning and AI and the tools of biostatistics and bioinformatics to be able to look at very large data sets that are part of looking at genetic variation in a person and comparing it to the genetic variations in people that have gone on to get disease or not to get disease. Use those algorithms derived from the machine learning and AI to inform then what an individual can expect in terms of their risk of having future disease. So these are extremely large data sets that are literally, if you think of it as a matrix, it's a million rows deep and, thousands of columns wide, and you're trying to find patterns in that data. And prior to just the last few years, that was impossible. Now with AI and massively parallel cloud computing and super fast computers to microprocessors, et cetera, this is coming on very quickly now. And remember that like Jensen Huang is the CEO of NVIDIA, predicts that this speed and computational ability is 1,000,000 times better now than it was just a few years ago, and will be 1,000,000 times better a few years from now. And when he says 1,000,000, he means 1,000,000, like 1 times 10 to the sixth, like 1,000,000 times faster, cheaper, etc. And with 5 to 10 years from now, 1,000,000 times faster than even will be 3 years from now. And so that's 1,000,000 times 1,000,000 faster than it is now. So this is going to get really crazy in our ability to analyze large data sets, and that's going to even further allow us to personalize individuals, understand individuals, and project their risk for chronic diseases and focus the tools of precision medicine on those people appropriately. So a little bit of a high school science review. On the left of the screen, you see the usual double helix. I say the usual, of course, it's always a double helix, DNA. And the rungs of the ladder that are made up of letters of your genetic code. So those letters are inherited from your mom and dad, 3 billion letters spread over 23 chromosomes from mom, 3 billion letters spread over 23 chromosomes from dad. Now, we're 99.9% the same, all of us speaking and listening today. And that means that of those 3 billion letters, there's 3 million or so of them that are going to vary from person to person. That 0.1% variation in the genetic code results in all the diversity. So to bring in a couple of terms, in the middle column there, you see the blue A here and the blue A here. That's what most people have, but this particular person has a G there. So that's, and to bring up a term, this is called a SNP, which stands for single nucleotide polymorphism. That's a lot of words there to say at this point at the genetic code, this single point, so at this one point, the nucleotide, which is just the letter of the code, is polymorphic, meaning it can take different forms. So you can have different letters. So SNP just means that point in the genetic code where people have variation. And so if you have at that particular point what most people have, we call that the major allele. If you have what fewer people have, we call that the minor allele. Now, this little one letter change in the DNA gets transcribed into messenger RNA. So we all hear now about RNA, thanks to the COVID virus. This RNA exits the nucleus and goes out into the cytoplasm, where it's translated into a sequence of amino acids. A single letter change here, though, will result in a change
in
of
the mRNA.
codon. And that single letter change ultimately can result in an amino acid substitution. In the case of sickle cell anemia, that single base pair change, that single SNP variant, can result in a pretty terrible disease called sickle cell anemia, where the cells, the hemoglobin and the red blood cell collapses in the face of cold temperatures or low oxygen tension or the like. Or it can be if you can have a SNP that results in someone tending to have blue eyes as opposed to brown eyes. So some of them, some of these SNPs are, functional or benign, and some of them can go on to cause disease. So understanding for an individual's collection of these points of variation, how that portends for the risk of developing a chronic disease in the future is predictable thanks to all that ability we have to analyze large data sets. Did you have a question?
Yeah, well, I just wanted to summarize from what I'm currently understanding in the reason why precision medicine and personalized medicine is so heavily weighted, not heavily weighted, but the help and assistance of being able to analyze all of these different data sets is significant to being able to understand a person's body and genetics based on, so like just from this presentation that you have right here, billions and pieces of data sets that just come in, and you're just trying to figure out how it actually works and is put together.
Yeah, like there's no way that you're going to have personalized precision medicine 10 or 20 years ago. In the way that our healthcare system evolved to be reactive, the so-called break and fix system based on insurance reimbursement, that's insurance being based on who you were employed by, et cetera. These are all kind of quirks of the 20th century. And I want to say, I'm sure people were just doing the best they can do. It's just that the time has come because of these massive advances in biologic science and the ability to handle and work with these enormous data sets due to advancements in computational ability and advances in data science and machine learning and artificial intelligence, combined with having sequenced the human genome. We're at an incredible inflection point where the way healthcare will be delivered in the future, I'm not saying it's probably going to be different. I'm going to tell you for sure it's going to be different. I'm going to tell you for sure almost every baby born will be sequenced at the time of birth, so that problems even in infancy can be anticipated and dealt with, and diagnostic dilemmas informed right off the bat. And then, of course, as the person gets into their adult years, it's going to be very important to be able to assess the risk of future disease and take actionable steps to mitigate those risks.
What it almost seems like, and this is such a dumb example, but I imagined that there was just this container that you just put salt in, and then you put pepper in, you put paprika, you put oregano, you put all of these different spices, and then you have this technology and computer that can go in and just analyze what is actually inside of there, given all these different spices, piece by piece by piece. You have all these different...
Yeah, and then take a machine learning tool based on that and project how most people would describe what that tastes like.
Yeah. Whoa,
you know what I mean?
Yeah, that's a great way to put it.
And to be able to do that, without firsthand knowledge, but just a predictive capability. And so you're really getting at it. So, personalized precision medicine, in addition to being proactive and personalized, but it's really what we're leveraging is the ability to predict. And if you can predict, that gives you a big advantage. And one of what I'm about to say has nothing to do with the slide that's up right now, but I like to think of it as like when you're in Las Vegas and you're playing blackjack and you count cards to try to decide when the dealer has a preponderance of face cards. Because if the dealer still has a lot of face cards in the deck and they've played all the little cards, then when they have that 5 or 6 up, they're going to bust because they're going to flip over. So people that count cards are carefully doing that. And when they think that the dealer has a preponderance of face cards, they increase their bet. And that's why they can beat the odds by counting cards in Las Vegas. So what we're trying to do is something similar. We're trying to assess where the risk is and where the risk isn't for a particular individual and push our chips toward those places where the risk is higher. And I'm sorry if I'm probably given that example on a previous.
No, actually, no, you haven't. That was a good example. That was the first time I've heard that. Great example. Okay.
So if you look, again, some basic genetics here. Many of us have heard about the BRCA genes that put a woman at risk for breast and ovarian and pancreatic cancer and put a man at risk for male breast cancer, prostate cancer, and pancreatic cancer. If you look down at the x-axis here, the horizontal axis, the allele frequency for BRCA is like 1 in 1,000 people has that allele. Now, we know that if you have that allele, that risk allele, we call it, the risk variant, or TP53, many people will have heard of that one, that puts you at significant risk of developing cancer. So the y-axis here, the vertical axis, is relative risk. So you can see people with the BRCA or the TP53 are 10 to 15 times more likely to develop cancer in those organs I mentioned than the average person. And so those allele frequencies are relatively rare in the population because, as Captain Obvious would point out, people that die young don't procreate as much. And so that allele frequency gets diluted through evolution. But did you know that there's a whole slew, and this is why cancer, for example, is so complicated, and slew is not a scientific word. I'll say a whole bunch, and also not scientific.
That was, yeah.
A ton of risk alleles that put you at slightly increased risk and are very common in our population. What's interesting is you can, again, through bioinformatics, and in the case of where this is right now, it's pretty simply looking at the risk of each individual allele and combining them via a weighted average to determine future risk of, in this case, breast cancer. So this emphasizes this point. Each of these common SNPs, risk alleles, add together in combination to pose a significant risk. So to beat this into the ground a little bit, you can have these single gene risk, like we talked about the BRCA or TP53, that combined with clinical factors such as, like we've talked about, the things that put you at increased risk for cancer, cigarette smoking, obesity, diabetes, alcohol consumption, etc. And if you calculate and control for those things, you can come up with a disease penetrance. What we say that is, if all this is present, how likely are you to get the disease? So I'll just make the number up that you have the BRCA and you're obese, and with just those two things together, you would have a 80% chance of getting the disease. That's why you see women sometimes doing prophylactic mastectomies and taking their ovaries out and the like. So that's monogenic, gene variants, relatively rare, combined with clinical factors like cigarette smoking. Over here, we can look at the polygenic risk. Poly is many genes, genetic genes. So you can have a number of genes that have increased relative risk, slightly increased relative risk, not like not like this where you have 10 or 12, but where it's just like, oh, maybe 1.25, so like a 25% increased risk, not like the 10 times increased risk. So you can then add in those same clinical factors, environmental factors, and project a lifetime risk score. So what would be ideal is to look at everything, look at your monogenic that, do you have the BRCA? Do you have the TP53? Which of these lesser risky genes do increased risk, but not severely increased risk? How many of those do you have in combination and together with environmental and clinical factors, your diet, your exercise, your smoking or not smoking, alcohol, no alcohol, stress, etc. How does all this add up together?
It almost seems like a yin and a yang between the two, because like with the monogenic risk, you're looking for that single gene variant, and then you're referencing and also cross-referencing on the polygenic risk side multiple variants and how they're interacting to come with and I guess
It's
create
not
those
really
people.
yin or yang, it's yin and yang in
Yin
this
and yang.
case. So
Okay.
you really want to look at everything. And so that's where uh you've heard me talk about Bayesian logic-based theorem, where generally you use additional information that then you conditionally apply to your previous assumption. So when you use that type of methodology in this, you can be much more powerful in your predictive ability. And I apologize for invoking biostatistical jargon there, just that some people will know what I'm talking about. And then but I think the explanation is also good. I'll give it a further explanation of Bayes' theorem. Sean Carroll wrote a book, the Big Picture. I think he gives a pretty good explanation of Bayes' theorem in there. It's like, suppose you're a senior in high school and it's May and it's around that time you got to figure out which girl you're asking to prom,
Mm-hmm.
and you want for the girl to say yes, and so of course, and you're deciding, between two or three different ones, or it could be a girl deciding what guy she's going to ask. Of course, I have two daughters, so we got to get this squared away here. But suppose you're walking down the hall and the girl you want to ask says hi to you. And so now you have to assess how that affects the probability of whether she would have said yes or no had you asked her. So in other words, is she saying hi just because she feels particularly friendly that day, or is she trying to drop some 411 on you that you might want to ask her to the prom, etc. So you have to weigh those probabilities. You have to come up with some probability of how you assess this new information and apply it to your prior credence, and use that information together, because all these additional pieces of information together are going to change how you make your decision. I know that's confusing. I'll just let it drop
It's
there.
no, it's almost like the multiverse. I mean, as dumb as that sounds, where like each decision that you make is going to come up with just a different universe at the end.
Yeah,
I mean, that's how I conceptualize that.
so there you go. And really what the name of the game here is we're trying to, assess risk so we know where to push the chips. We're back to counting cards. And so this is kind of a fun depiction of predicting colon cancer risk. So along the left here, and so we're going to call this aqua colored dots here on the left. If you have no first degree relatives, so when I say first degree relatives, I mean mom, dad, brother, sister, your risk is a little less than the average. So a incidence of 1, or a relative risk of 1, is average. If you have no first degree relatives, your risk is slightly less than average. If you have one first degree relative, you're up here around 1.25 or 25% increase. So you're at slightly increased risk. And if you have two first degree relatives, you're all the way up here at 1.75, still less than twice average risk, but 75% increased risk. Now, if you look at this 359,000 people that had no first degree relatives, but you looked at their polygenic risk scores. So in other words, you are now adding information together. So you're looking at those polygenic risk scores that we talked about, those multiple SNPs that each particular one doesn't really do much to increase your risk, but how they combine together could possibly increase your risk. You're able to get more granular with your risk stratification here. So this 359 used to all be kind of clustered here at below average risk, when it really turns out that there's a subset that are at very low risk, some that are about where you thought they were, but then there's a subset that are at markedly increased risk, and increased even more than people that had a first degree relatives. Remember, this is just getting risk stratifying these people that had no relatives. Here over on the right in the blue, you do the same thing, looking at the bottom, middle, and top quintile of patients. You can see that you can again get a much more information. So this 42,000, it turns out that some of them are at very low risk. Some are about where you thought they were. But then there's the subset that are at markedly increased risk, even more increased risk than someone that had two first degree relatives. So this is how you use bioinformatics to assess risk. This is how we're using polygenic risk scores and integrated risk scores to see who's at risk for what, so that we can not treat them at that population scale, one size fits all. Does that make sense?
Yeah, and so what you're really comparing then is just you're able to see the risk that you have the further you go down into the family history of people that, let's say, did have it then. Is that correct?
Right. So family history allows us to do a certain amount of risk stratification. But then what I'm showing you is that you add into that, in addition to looking at whether or not they had a first degree relative, when you add in the knowledge of what these polygenic risk scores are for an individual, you can further risk stratify. You can not only, you can take, especially like this set of people that look like they're at about average risk, but actually some are much lower than average and some are higher than average. And that information is very helpful to a healthcare provider in deciding whether or not you should wait till age 45 to get colon cancer screening. Maybe you want to jump the gun on that colon cancer screening and get it at age 35. Or maybe you're this person out here with one first degree relative, but really you're at 1.75 relative risk, or 75% increased risk. Then maybe instead of every 5 years for your colonoscopy, you might want to discuss with your GI doctor and decide you're going to do it every 3 years, like you do if you happen to have polyps on an exam. Then they say, come back in 3 years. And you say, yeah, I have one relative with colon cancer. They'll say, oh, come back in 5 years then. And you go, yeah, but I did this additional testing and it said I'm at much higher risk than what you would expect just based on that. And they go, oh, well, let me see if there's science behind this. And then they may advise you to get screened every 3 years instead of every 5. Does that make sense?
Ah, okay. It like finally clicks then, and now and how it's relative to just this graph in each of the different categories that I'm looking at. So like
the bottom line is more information when thoughtfully applied can help you do risk stratification, which when we say that is we try to put people in categories that would identify them as being at much higher risk than you would have initially thought, or much lower risk. So knowing who's at risk tells you how to spend your industry, so to speak, your time and resources in terms of your healthcare to prevent a terrible disease like colorectal cancer. If you're not screened at the appropriate interval, you could develop advanced colon cancer, and that is very lethal. If you detect it early, when it's precancerous polyps or stage 1 or 2 and even 3 colon cancer, it's highly curable. Once it's stage 4, it's not it's not nearly as easily cured. Does that make sense?
Yeah, makes sense. And then just for my clarification too, the
Well, I don't mean cured. I mean that your survival chances are much lower.
Are increased.
Yeah,
right. So just for my clarification, on the left column, then that is not having a gene type. That's just looking at with or without family history. And then the middle is gene type with no family history.
What's in the middle is just this dot exploded out when you consider polygenic risk scores.
Gotcha. Okay.
And these 3 blue dots are just this dot exploded out when you consider polygenic risk scores. So this is just illustrating the power of using, actually, and this is polygenic risk scores and other clinical risk factors, but these are integrated risk scores, but polygenic risk scores. Actually, let me back off that. These are just family history plus polygenic risk scores.
Gotcha. And that's how you can get your most accurate depiction then of figuring out
most accurate depiction of the risk, so you can make informed decisions.
Got it. Okay.
So much better. So this slide, you have 1 in 400 women. Looks like guys to me, but I never noticed that.
Okay.
But it's adults, men or women, will carry the BRCA1, and wouldn't it be, and that's helpful, that we know that for that one woman in 400 that has the BRCA gene, that's helpful to know, and it will inform clinical decision making. But wouldn't it be so much better if we could catch the 80 people out of 400, the 1 in 5, that you can identify them at being markedly increased risk by looking at those common genetic markers, each of those less dangerous SNPs that when combined together, and when you consider other things like obesity, whether or not they've had kids, when they started their period, have they been on hormone replacement or birth control, add all these factors together and do an integrated risk score and decide who's at increased risk, and enroll them in high risk breast clinics, just like we did this one person here with the BRCA1 gene. Does that make sense?
Yeah.
This allows us to find many more vulnerable people. And we can do this not just with breast cancer or ovarian cancer, but with a number of chronic diseases that there's this capability for, and this capability is growing and growing every day. So there's and there's very strong science behind polygenic risk scores these days that published in Nature Medicine. There's a recent article, are polygenic risk scores ready for prime time? And the answer is yes, they are. So again, if you look at clinical risk factors together with polygenic risk, so this particular company will give you risk assessments on these 3, 6, 8 diseases in a woman and 7 in a man. Instead of breast and ovarian, they do prostate cancer. These are the things that are going to kill you. These are the horsemen, some of the horsemen we talked about. Type 2 diabetes, are you at risk or not? Coronary artery disease, are you at markedly elevated risk? This is currently the leading cause of disease. Cancer is going to overtake it this year, but we want to know about these more common cancers. It would be nice if lung cancer was on here. The good news is, though, that with lung cancer, I expect they will have this someday. But with lung cancer, luckily, there's a relatively straightforward screening exam that will catch lung cancer at an early stage. And that is a low radiation dose CT scan of the chest. People should avail themselves of that. It turns out only 1 in 20 people that are eligible for that test get it. So that's kind of a problem too. Another way that we can leverage knowledge of your individual genetics. So when drugs are designed and marketed and put out there, they're put out there for the average person that has the major allele, the so-called normal gene, and with the normal variant. And if you're one of the people that has the normal gene and you take the drug, you're going to have success with that drug. But if you happen to have a variant, and the gene they're talking about here would be like a gene that in the liver cranks out an enzyme that metabolizes the particular drug. So let's say that the drug is, I don't know, Prozac, and say for example, if you have this gene variant that is slower than average to metabolize Prozac, then you're more likely to accumulate the drug to higher levels than anticipated, possibly even toxic levels, and you would have a drug failure. So what this slide, not my slide, but whoever made this, I pulled it off the internet. If you consider pharmacogenomics and you select the right drug at the right dose based on the individual genetic variants, you can then have a successful outcome, even though the person has a SNP variant, just by making adjustments in either the particular drug that you're selecting or how you dose it. Does that make sense? Any questions there?
Yeah. So, I mean, if I were to just summarize it, then pharmacogenomics, it takes into account how your body reacts to the medicine, but down to that, let's say, on the genetic level then.
Right. So
oversimplification of it, but
yeah, no, so there's 20 or 30 of these, they're called cytochrome P450 genes. So there's like 20 or 30 genes that together, or 20 or 30 genes, and the gene product being the protein enzyme, the enzyme. So 20 or 30 different enzymes that break down most of the drugs that we take. If you happen to be taking a drug that the major metabolizing is going through an enzyme that you have that variant, it doesn't work very well, then you're not going to have you're not as likely to have success with that drug. Does that make sense?
Yeah, so the drug isn't going to be as effective then if...
Well, it might even be toxic. So if it's slow to metabolize it, you're going to accumulate that drug in your bloodstream to much higher levels than was anticipated, because when the drug dosing, et cetera, was done, it presumed that you had normal gene function there. But these variants in the enzymes that metabolize drugs are very common among people, right? The average person, out of those 20, 5 of them, they're either going to be metabolizing the drug way faster than anticipated. And in that case, you're taking the drug and it's getting metabolized, and it's never at a therapeutic level in your blood. Whereas if you have a variant that is very slow to metabolize it, then you're going to accumulate greater levels in the blood than you would have predicted based on the normal variant. And that's not good either, because that can lead to drug toxicity.
So let's use an example. Let's say I was allergic then to erythromycin. Would knowing this then and knowing this information be able to streamline a better drug that they would know, so I don't have to go through the process of getting hives from erythromycin? And then...
Yeah, well, The short answer is ultimately that capability will be there theoretically. Right now, that most of the quote allergic reactions we're talking about would be disastrous outcomes if you took erythromycin beyond hives. Would be like it gets for certain people. I'm making this up, but just for an example, but erythromycin might accumulate to toxic levels and cause not just hives, but literally cause like a condition called Stevens-Johnson, where your skin just dies and falls off your body on sheets.
Yeah,
So,
geez.
there, this type of um, analysis and looking at gene variants, and again, they're going to, and we don't have this right now, but I would say what at the rate that AI and data science is going, I would anticipate that we would have exactly what you're talking about certainly within 5 or 10 years.
Wow.
But this is the first foray into that in terms of the rate that drugs are metabolized.
Understood.
And certainly there are some drugs that you can look at a person's genetics and know that person should never get that drug because they are at increased risk for an allergic reaction or some horrible reaction. And there's a handful of those and that we're able to predict. But I think in the future, that fund of knowledge, that body of knowledge will be much larger. So this just depicts that. So the green normies there, they take the drug and everything's fine. The orange guys, they take the drug and have a toxic response because they accumulate too much drug. And then the blue people, maybe they metabolize it too fast and they don't respond at all to the drug because they never really mount a drug level, because it's just metabolized within minutes after you take the drug. So Biography Health, the what do we call them? The sponsors of our show, the great sponsors, have built an app that allows physicians, providers, people like me to bring all this information together in a way that facilitates decision making, clinical decision making. We call it clinical decision support. So for things like vascular diseases, we can look at if there's any monogenic variants, like a common one in the vascular world would be one called ApoA2, where it's a form of familial hypercholesterolemia. We look for the presence or absence of that. I think we were discussing this with you. And then the polygenic risk scores, what are those SNPs with increased relative risk, but not markedly increased relative risk? What happens when we look at all those together? And we can look at how you compare to other people in terms of your risk for disease. And then we also feed into this same column not shown here. We call this the medical Netflix user interface, because there's an infinite number of columns going left to right, and there's an infinite number of rows going top to bottom to put all this information in. So in addition to monogenic risk, polygenic, or here's the single gene risk and polygenic risk, but we also look at current lipid levels. We look at your family history, your smoking history, all these things that would influence your risk for coronary artery disease and stroke. And we assess that risk. And if it's, depending on what it is, we'll drive certain clinical recommendations like, hey, you're at markedly elevated risk of coronary artery disease, even though you're only 40 years old. I would have your coronary arteries looked at because there's relatively inexpensive tests out there, and it does require a CT scan with contrast, but you can answer that question. I'm only 40, but my risk, as informed by these bioinformatics, et cetera, tell me I'm at increased risk maybe I should take a look and decide if I should be doing something about it. And why do we do all this? Because the easiest way to live longer, we all want to live longer in that extended health span where we're at high functional capacity up till the day we die. Well, the easiest way to live longer is not to die prematurely of something preventable.
Thanks for joining us on the Precision Medicine Made Simple podcast. If this episode inspired you to learn more about your health, please leave us a review and share the podcast with a friend. Your feedback helps spread the word about the power of precision medicine. Remember, taking action today can lead to a healthier tomorrow. See you next time.